Today, in this second part of e-learning session, we will discuss about our next phase, that is M phase. In the previous lecture, we discussed about cell cycle, and we discussed that in a cell cycle there are two phases. One is interphase and another one is M phase. First, the genetic material, it becomes duplicated, it becomes replicated, and then the segregation of this genetic material take place in the M phase. Now, this M phase can be mitosis or it can be meiosis. This M phase is the most controversial and the most studied phase. Now, the picture that you are seeing is just 3% of a single molecule of DNA of one of the chromosomes of human beings, of a human diploid cell, and you can imagine that how difficult it is to replicate this enormous amount of DNA. So, from that figure, we can say that the process of first replicating the DNA and then segregating it into two parts is really a very difficult task for a cell. Now, today in this M phase, we will discuss about the first type of division, that is done by every cell, which is mitosis. Now, before looking into the details of mitosis, we will discuss some common terms. For example, chromosome. Chromo means colored and zo means body. Now, this is the condensed chromatin or genetic material that is visible during the M phase. Now, another question, what is chromatin? Now, chromatin is unraveled condensed structure of DNA which is meant for packaging. If we start with the DNA like this is DNA. When it binds with the histone proteins, it becomes chromatin, and when it condenses, then it becomes chromosome. So, chromosome is a condensed genetic material which is visible or which is present in the M phase, and chromatin is the relaxed stage of genetic material that is present in the interphase. M phase starts with the condensation of chromosomes. The first thing is the condensation of chromosomes. Second thing is spindle. Now, what is this spindle? Spindle apparatus is meant for the equal and proper distribution of chromosome, and they are produced before condensation. The spindle apparatus, it appears before condensation is complete. It is uniquely present in eukaryotes. It is uniquely present in eukaryotes. Of the spindle is a central mechanism for the distribution of genetic material in the daughter cells. Spindle not only distributes the genetic material, but it also aligns the chromosomes properly. Two abnormalities that are associated with the condensation of chromosomes and spindle organization are: first is endo-reduplication, and the another one is endomitosis. For understanding these two abnormalities, Let's start with GO, which is the stage when cell is not going to divide. If it gets a signal and it enters into the interface, that is, it is going to divide G1 S G2, then coming to P, yeah, that is prophase. Now these are the different stages of interface G1, S and G2. Now endo-reduplication abnormality is observed when the signal for chromatin condensation as well as signal for spindle organization are absent. So this abnormality, endo-reduplication, it appears when there is neither signal of condensation nor signal for spindle appearance. Whereas endomitosis is observed when the signal for spindle organization is absent. Now endo-reduplication, it results into polytiny chromosomes, whereas the endomitosis, it results into polyploidy. Now this was just very few things about the spindle and chromosomes. Now coming to our first division, that is mitosis. Can be defined as any type of division in which the chromatin material is first doubled and then it is divided equally in two daughter cells, and then it is divided into equal daughter cells. So this is what mitosis is. The daughter cells and the mother cell, they have same number of chromosomes, there is no change. The mitosis can be divided into following stages. Prophase, metaphase, anaphase and telophase. Now we can study mitosis in two phases. The first is karyokinesis and the second is cytokinesis. Karyokinesis, the division of nucleus. Cytokinesis, the division of cytoplasm. As I already told you that there are four distinct stages in mitosis: prophase, metaphase, anaphase and telophase. We have we can further divide prophase into three more stages: early prophase, middle prophase and late prophase. Though these phases are not very discrete, pro, as a name it indicates, pro means beginning. The chromatin, it starts condensation and the chromatin starts taking shape as chromosome. The nuclear membrane, this is the nuclear membrane and this is the nucleolus. They are still present in prophase. We can distinguish prophase with metaphase with the presence or absence of these two things, nuclear membrane and nucleolus. The time they disappear, prophase is no more, but in some primitive plants and animal species, the nuclear membrane and nucleolus, they do not dissolve even after prophase. So in this prophase, the chromosomes, they start visible, they start appearing. Cell increases in its volume, the size of the nucleus it increases, the nuclear membrane are present, the chromosomes are long, uncondensed and uncoiled. In the middle spindle they start appearing, the chromatids they start becoming shorter and thicker. In the late prophase the chromosomes they become very clear, most condensed and coiled, and mostly the nuclear membrane and nucleus they disappears. The chromosomes at this stage they start attaching with the spindle fibers. The speed of movement of the chromosomes is about 0.01 to 0.05 micrometer per second. Now, this structure that you are seeing here is a duplicated genetic material. The genetic material which got duplicated or which got replicated in the interface. This single thread is not single in nature, it is like this, and these two fibers are attached at a place which is made up of protein cohesin. The chromosomes are condensed. This requires ATP and a protein complex called as condensin. For condensation, condensin is required. Then the chromosomes they separate and distribute equally in two daughter cells. Now, there are some other things that are associated with the prophase here. Firstly, in animals, centrosome is present. A centrosome is an organelle which has centrioles, and these centrioles they move to opposite poles like this. The spindle fibers they appear in the cell. The spindle fibers, what are these? These are the microtubules of 250 angstrom in diameter. The place of the chromosome with which it is attached with the spindle is known as primary constriction. This centromeric region is made up of protein along with centromeric DNA. Now, the spindle has got two poles. Spindle is of two types, astral and unastral. Astral, in which aster fibers are present, and anastral, where the aster fibers are absent. The cases where the centrioles are absent are called as acentric, and where the centrioles are present is known as centric. In this metaphase, the relative position of chromosomes on the spindle equatorial plate is more or less fixed. This is a linear distribution of the chromatin, as the distribution is non-random. The bigger chromosomes they occupy the peripheral part, whereas the smaller chromosomes they are in the center. In metaphase, the chromosomes they become more short, more condensed and more coiled, and this is the imaginary metaphysic plate on which they are attached. Now here again two things are there. One is known as amphitallic condition and another is known as syntelic condition. Now amphitallic is the wild condition in which one kinetochore is attached to one spindle and another kinetochore to another spindle. It is present in both mitosis as well as meiosis. The syntelic condition is like this, where both the kinetochores are towards one side and it is present in only meiosis. So syntelic condition in meiosis and amphitallic condition is in both mitosis as well as meiosis. Now coming to anaphase, where these two sister chromatids, they start separating from each other. The centromere splits. The anaphase is of two types. The movement of chromatids is of two types, anaphase B and anaphase A. The movement of a chromatid towards the pole is known as anaphase A movement, whereas the separation of these two sister chromatids is known as anaphase B movement. The speed of anaphase chromosome is about 1 micrometer per second. This can be stopped if we apply a force of 7 into 10 to the power minus 5 dynes. Then the chromatids can be stopped. Number of factors may be responsible for this movement of chromatids, as the contraction or shortening of the spindle fibers, or due to some viscosity changes in the cell, or due to some electrostatic forces in the cell. This movement of chromatids is known as congregation. And as we can see in the last stage, the chromatids, they come to lie in opposite poles. They become again relaxed, they become uncondensed, long. Both nuclear membrane and nucleolus, they reappears. So in this way, the karyokinesis is complete. Two nuclei formation take place. Now coming to the next stage, which is known as cytokinesis. Now the cytokinesis can be by two methods. First is known as cell furrow method, which is very common in animals, and the next is cell plate method, which is very common in plants. In cell furrow method, the cell divides from outside to inner side. Furrows appear, which becomes deeper and deeper, and finally both the cells, they separate from each other. In cell plate method, from the center, the cell plate, it extends towards the periphery, making a plate-like structure. This is due to endoplasmic reticulum vesicles, which collects in the center, thereby separating two cells. And thus, this is how two cells are formed from a single cell. Now, today in this e-learning session, we learned about a very simple division known as mitosis, which is responsible for growth and which is a very usual phenomenon. In the third part of this cell division, we will discuss about next division, that is meiosis. Thank you.